Hello students, I welcome you to EPG Pathshala. My name is Dr. Shivkumar Sood. I am senior scientist in the Department of Animal Biochemistry at National Dairy Research Institute, that is ICAR NDRI in Karnal, Haryana, India. Today I am going to talk about protein structure hierarchy from the paper Biostatistics and Bioinformatics. Dear students, proteins are the most important biomolecules that control important life functions such as enzyme catalysis, storage, coordinated motion, mechanical strength, and many more. The most important function begins with the mechanical support provided by proteins in the biosystems, that is the cells. This is provided by the protein collagen. This is requiring energy and other support which is provided by catalysis, that is performed by the enzymes which are proteins. Then storage of the molecules like storage proteins, transport proteins, ferritin, transferrin, and proteins involved in coordination or generation of the nerve impulses, that is enzyme acetylcholine esterase, and then immune protection by antibodies which are again proteins. So these are the important life functions which are provided by the proteins. So therefore today we are going to talk about protein structure which is achieved through hierarchical manner. The hierarchical manner here means that this structure is achieved in a seriatim or sequential manner. That is, first the structure achieved at the current level, then the next level, followed by the next level. If the structure achieved at the current level is not correct, then the structure achieved at the next level will again be not correct, and therefore the final structure which is achieved will not be able to perform its function. Therefore, the structure to be achieved at the current level is to be correct. There are 4 levels in the protein structure hierarchy. These are called primary level, secondary level, tertiary level, and quaternary level. At the first level, the linear sequence of amino acids joined together through peptide bond establish the primary level. That is the primary structure, and this provides the basis that the linear sequence of amino acids joined together through peptide bond provides the specificity to the protein itself. At the next level, that is the secondary level, the level achieved is through only hydrogen bonding of the main chain. In the primary sequence, there are 2 components. One is the main chain, that is nitrogen, C alpha, then carbonyl carbon, then again nitrogen, C alpha, carbonyl carbon, again nitrogen of the amino group, C alpha, carbonyl carbon, that is nitrogen, carbonyl carbon, C alpha, carbonyl carbon. So this is called the main chain. To the C alpha, that alpha carbon, side chains are attached. These are known as side chains which are different in different amino acids. There are 20 naturally occurring amino acids, and the side chain is different in each of the naturally occurring amino acids. The main chain provides amide bond, that is the peptide bond which has carbonyl oxygen and nitrogen. These 2 groups are the hydrogen donor and hydrogen bond acceptor groups. Therefore, these provides bonding for the main chain groups of the carbonyl oxygen and the amide nitrogen. They form the hydrogen bonds among themselves. This is what we call secondary level. Therefore, the level, that is the secondary level structure, is achieved through formation of only hydrogen bonds of the main chain atoms. As in the primary sequence, the level achieved is through the formation of the peptide bonds. These are covalent bonds, but here these are non-covalent hydrogen bonds. And these provides a link to fold the primary sequence through this level, a link to the next level, that is the tertiary level. In the tertiary level, the interactions are both covalent and non-covalent. In the non-covalent interactions is the electrostatic and hydrophobic, hydrogen bonding, salt bridges, and the covalent includes the disulfide bonds between the cysteine residues. So these interactions are not from the main chain. These interactions at the tertiary level are among the side chains only. Therefore, we have now the third level. From the primary mean peptide bond, secondary mean main chain atoms interacting through hydrogen bonds, and then tertiary side chains interacting through covalent and non-covalent interactions for the folding of the peptide chain or the polypeptide chains. This level has two regions. First is the core, which is the tight packing of the secondary structures achieved at the secondary level. These are alpha helices and beta sheets which are packed within the core, tight packing, and the forces involved are mainly hydrophobic. That is, no hydrophilic interactions inside the core. Then the second region is surface. So we have a core, we have a surface at the tertiary level. Core is tight packing and the amino acid side chains packing is involved through the exclusion of water from inside the core, mainly through hydrophobic interactions. And then the surface, which is mainly hydrophilic. So the secondary structures, which are alpha helices and beta sheets, they are packed inside. And the amino acid side chains inside the core of the protein are mainly hydrophobic, whereas the amino acid side chains which are present on the surface are hydrophilic. This core provides a scaffold onto which the hydrophilic or the surface loops are arranged. These loops provide the active sites of the enzymes or the binding sites of the receptors. This scaffold, for example, let us say this is the protein which has two lobes. This is one lobe, this is second lobe, and this is the surface cleft which provide the active site for the enzyme or the binding pocket for the receptor, say for some hormone. So this scaffold provides the surface clefts in which the residues are mainly hydrophilic. This is what we call the tertiary level, and the primary chain or polypeptide chain which is able to fold independently is called a domain. A complete chain may fold into single domain or it may fold into two domains, but in that case the two domains must be folding independently. Let us say that we are having a protein with 200 amino acids, from amino acid 1 to amino acid 100. If we cut between peptide bond 100 and 101st amino acid and fold these two separately, if they are able to fold independently, then they are called domains. If let us say that from amino acid 1 to 10 and then from 101 to 110 they fold together, then they may form two lobes, but they are not called domains. So an independently folding unit is called a domain, and a protein may have a single domain or may have more than one domain. Then at the next level, domains or what we call the subunits, a protein having two lobes is called a subunit. They interact with each other through non-covalent interactions, so that they form what we call the fourth level, the quaternary structure. This level of structure is achieved mainly for the regulation. Say the best example is hemoglobin, which is having 4 subunits, 2 alpha globins and 2 beta globins. They interact to each other through non-covalent interactions and help in regulation of the binding of the oxygen. Otherwise, you see the binding is always hyperbolic. But once the protein quaternary structure is coordinating with each other, then this hyperbolic carb binding changes to sigmoidal, that is cooperative. Mean first oxygen will bind with difficulty, but the last oxygen will bind very easily. This is what we call cooperation or cooperative binding, and this is achieved through this level. So we are having 4 levels. Primary level achieved through peptide bond formation, covalent interaction only. Secondary level, main chain hydrogen bonding. Tertiary level, side chain interactions including both covalent and non-covalent, and quaternary level interactions amongst the subunits or the domains, and then this is through only non-covalent interactions. This domain structure is actually achieved during evolution to combine the functions together. For example, we are having epidermal growth factor. This is having a single domain. Then we have serine proteases like trypsin, chymotrypsin. These are having 2 domains. Each domain is coming independently. Mean during evolution, a domain is evolving because it is able to fold independently. One domain from one protein may combine with another domain from another protein. This adds to the functionality, combining together the functions, and this is how the evolution has taken place. Therefore, the objectives in this module are to learn protein structure hierarchy at 4 levels. First level involving joining of amino acids through peptide bonds to form a linear polypeptide chain to achieve primary structure. Then at the next level, regularly repeating structures are formed through hydrogen bonding among main chain peptide bond atoms to achieve secondary structures. Next level involves formation of independently folding subunit or domain structure by regularly repeating structures joined through irregular loops, involving interactions of the amino acid side chains to achieve tertiary structures. At the last level, subunits oligomerization occur through non-covalent interactions to achieve quaternary structure. Proteins are linearly arranged sequence of naturally occurring 20 amino acids through peptide bonds. Theoretically, the naturally occurring 20 amino acids can be arranged in enormous number of sequences. For example, say there are proteins having only 2 amino acids. We are having 20 amino acids. If we are having protein with only 2 amino acids, then it means that these 20 naturally occurring amino acids can be arranged into 20 raised to the power 2 sequences. That is 20 into 20 means 400 sequences. That is dipeptide. Mean naturally occurring 20 amino acids, if arranged as dipeptide proteins, then there will be 400 proteins. Let's say that we are having proteins having length of 3 amino acids only. Then it will be 20 raised to the power 3. 20 into 20 into 20 means 8,000 proteins. 20 raised to the power 4, that is 20 into 20 into 20 into 20 means 1,60,000. So in this way, let us say that we are having linearly arranged sequences and we want to calculate how many proteins will be available theoretically for the natural proteins. On an average, the natural proteins are having amino acids around 100. 100 amino acid in naturally occurring proteins. So the number of protein sequences will be theoretically possible will be 20 raised to the power 100, which is enormous, huge. Whereas naturally occurring bacteria like E. coli harbors only 3,000 proteins, and complex mammalian system harbors around 30,000 proteins only. So this means that only some of the sequences are allowed. Rest are not possible because of the maybe steric clashes or non-performance of their function. So to achieve specific function through evolution, the nature has selected only some of the enormous number of possible sequences. For example, 30,000 in complex mammalian system. So therefore, through evolution, only those proteins which can actually perform some function are selected, rest are not evolved. An amino acid has 4 groups attached to a central carbon in tetrahedral conformation known as C-alpha. The central C-alpha atom has 3 common groups attached in each of the amino acids. The amino group is bonded in direction going below the plane of the paper, shown with dashed wedge in this slide. Second group is a carboxyl group bonded in direction going above the plane of the paper, shown with solid wedge in this slide. Third is a hydrogen atom, shown with normal line to convey that it is bonded in the plane of the paper. The fourth is a variable side chain in each of the 20 naturally occurring amino acids and is shown with the normal line to convey that it is also bonded in the plane of the paper. There are 20 naturally occurring amino acids. At the root level, the amino acids can be classified in 3 groups. First is unique group having amino acids glycine and proline. Glycine has attached only hydrogen as the side chain, and proline is attached with 3 methylene groups chain attached to both C-alpha and nitrogen of the amino group to form an amino acid starting with I as opposed to A, amino acid versus amino acid. The second group has polar side chains either uncharged at neutral pH or side chains having charged acidic or basic groups. The third group has non-polar side chains either aromatic or hydrophobic. The second group with polar side chains has sulfur containing amino acids methionine and cysteine. The second group with uncharged hydroxyl group at neutral pH include serine, threonine and tyrosine. The second group also includes amide group containing amino acids asparagine and glutamine. The second group with polar side chains having charged acidic group include aspartate and glutamate. The second group with polar side chains having basic groups include histidine, lysine and arginine. The third group having non-polar aromatic side chains are present in phenylalanine and tryptophan. The third group having non-polar hydrophobic side chains may be slightly bulky as in leucine or isoleucine, or small side chains as in alanine and valine. The 20 naturally occurring amino acids are linked together through amide bond known as peptide bond. For forming a peptide bond, the hydroxyl part of the carboxyl group in combination with hydrogen from amino group of another amino acid is removed as water. This forms a dipeptide having one peptide bond. This is beginning of achieving the primary structure. This dipeptide with side chain R1 and R2 may be linked to third amino acid with side chain R3 through the formation of second peptide bond. Linking third amino acid with side chain R3 forms a tripeptide. Linking together several amino acids through consecutive peptide bonds gives a polypeptide. PepDraw is an online tool which allows drawing any polypeptide. Simply click on amino acid buttons to create a polypeptide as shown for amino acid sequence here in this slide and click Draw Peptide button. PepDraw reports computed peptide properties such as mass, isoelectric point, net charge, hydrophobicity, and other properties. It also displays its primary structure with amino acid side chains as shown for generated polypeptide. The sequence of a polypeptide is always written from N terminal to C terminal. The main chain atoms include nitrogens of the peptide bond, including N terminal, C alpha, carbonyl carbon of the peptide bond, and the last C terminal carboxyl group. The main chain atoms are shown here in light green background for the above peptide. The side chains are attached to C alpha atoms. The side chains are shown in light gray background for this peptide. The polypeptide sequence contains linked together amino acids in which the side chains are away from each other in three-dimensional space. However, in this extended form, amino acid side chains are not able to work in a coordinated manner. To bring together side chains on different amino acids in the sequence, the polypeptide chain folds with rotation around single bonds in the main chain. This brings the main chain atoms placed at short or even at longer distances in close proximity with each other and helps acquire structures able to work in a coordinated manner. The folding of polypeptide chain with rotation around single bonds in the main chain so as to bring the peptide bond atoms closer to provide hydrogen bond donor and hydrogen bond acceptor groups for the formation of hydrogen bonds. The atoms linked together through single bonds are capable to rotate around their single bonds. The atoms may rotate either clockwise or anti-clockwise. The rotation around single bonds in diatomic molecule is not meaningful. However, it becomes meaningful in polyatomic molecules with other atoms attached to these two atoms. Let us say that each of the two atoms, say atom number 2 and 3, has attached single atom to each. Say atom number 1 attached to atom number 2 and atom number 4 attached to atom number 3, as shown here in this slide. The angle between the atom 1 and atom number 4 in this example is called dihedral angle, and its value in this case is 0. This is due to all the 4 atoms lying in the same plane. With rotation around single bond between atom number 2 and 3 in clockwise or anti-clockwise direction, the atoms attached to atom 2 and 3 will also rotate to occupy new positions as shown, and the angle, that is the dihedral angle between atom 1 and 4, will change to the extent of rotation in clockwise or anti-clockwise direction. Although there is a possibility of rotating a bond through 360 degrees in either direction, but 180 degree rotation in clockwise direction is represented with positive sign, and 180 degree rotation in anti-clockwise direction is represented with negative sign for rotation. However, the outcome is the placement of atom number 1 and atom number 4 in 3-dimensional spaces which are different for each of the atom. The 4 atoms on paper can be represented in this manner, where normal bonds represent atoms in the plane of the paper and solid wedge-shaped bond represent atom above the plane of paper. In polypeptides, the main chain atoms include amino group nitrogen attached to C alpha, followed by C alpha attached to carbonyl carbon. This is repeated for the next amino acid with amino group nitrogen attached to C alpha followed by carbonyl carbon. This regularity of atoms repeat the atoms along main chain as amino group nitrogen, C alpha and carbonyl carbon. The tripeptide in this slide shows 2 peptide bonds number 1 and 2. The analysis of the protein structure shows that normal carbon nitrogen single bond length is 1.49 angstroms and the normal carbon nitrogen double bond length is 1.27 angstroms. However, peptide bonds with linked carbon and nitrogens has bond length equal to 1.32 angstroms. This carbon nitrogen bond length of 1.32 angstroms is in between single and double bond of the carbon nitrogen. This shows that the peptide bond is having partial double bond character and therefore all the 4 atoms, that is oxygen, nitrogen, carbon, hydrogen in the peptide bond are in a plane as shown by dotted line boxes in this slide. With the partial double bond character of carbon nitrogen atoms in main chain, the rotation around these bonds is not possible because the rotation is possible only around single bonds. Therefore, rotation of bonds between only peptide nitrogen and C alpha as well as C alpha and carbonyl carbon is possible. The rotation of bond between peptide nitrogen and C alpha results in dihedral angle or also known as torsion angle and is named as phi and represented with the symbol phi. Similarly, the rotation between C alpha and carbonyl carbon results in a dihedral or torsion angle named with symbol psi. However, the rotation between carbon and nitrogen atoms of the peptide bond is not possible and the angle is represented with symbol omega. The angle omega in proteins is always in trans configuration in geometric isomerism and therefore is approximately 180 degrees. Although all the possible values for phi and psi, that is from plus 180 to minus 180 are available, but yet theoretically these available possible values are not utilized in the proteins. In proteins, what has been observed that there are 2 sets of phi and psi angles present in majority of the proteins. The first set is having phi value minus 60 and psi value minus 45. This folds into what we call alpha helical structures, that is a spiral coil. This coil, what we call alpha helix, is having this set of values, that is minus 60 for phi and minus 40 for psi. The second set commonly observed in the natural proteins is having value 5 minus 120 and psi plus 120. These phi and psi angles are repeated, mean for each of the amino acid present in alpha helix or beta strand will have this set of value only. That is for alpha helix, every amino acid present in a alpha helical secondary structure will have the value for phi minus 60 and for psi minus 45. So, similarly, for the beta strand, it will again always will be fixed approximately minus 120 and plus 120 in beta strands. In the helix shown here, only main chain atoms are shown. In this structure, each peptide nitrogen is shown as blue, C alpha as black and peptide carbonyl oxygen as red. In alpha helix, every current amino acid is hydrogen bonded to 4 residues ahead, that is every 5th residue in the chain. This helix is classical alpha helix. The alpha helix represents that the word alpha is the discovery of a first regularly repeating secondary structure which is a helix. Every backbone NH group of the 5th amino acid needs a hydrogen bond to the backbone CO group of the first amino acid. Consequently, every current amino acid is hydrogen bonded to 4 residues away, which places CO group of the current amino acid exactly in line with the NH group of the amino acid number 5. The helix is formed by 3.6 residues per turn. This corresponds to 100-degree turn per amino acid residue. Each residue advances about 1.5 angstroms in length. This results in the length of 5.4 angstroms per turn of the helix, which is the product of 3.6 and 1.5. Therefore, helix structure is repeated every 5.4 angstroms. Certain amino acids such as leucine, methionine, glutamine favor formation of alpha helix, whereas others such as valine, serine, aspartic acid and asparagine tend to destabilize helices. The side chains in helix protrude out of the helical structure. That is, they never come inside the helix. In alpha helix, every first amino acid is hydrogen bonded to the next fifth amino acid, that is 4 residues ahead. Sometimes in proteins, the helical structure is formed by the hydrogen bonding between the first amino acid and the fourth amino acid, that is the 3 residues away from the current one. This is having 10 atoms from the first to the fourth residues. So, since this is bonded to the 3 residues away with 10 atoms in between, this is named as 310 helix. And in this helix, the phi angle is again minus 60, whereas the psi is not minus 45 but minus 30. So, this 310 helix is present in the natural proteins, and these helices, 310 helices, they occur normally at the N-terminal or at the C-terminal end of the proteins. In addition, helical structures are having a skew sense. Skew sense means that how they are moving, whether this movement is from the right to left or from left to right. For example, in alpha helix, which is a spiral coil like this. So, whether this is moving in this direction or in opposite direction, this is what we call a skew sense. If the helical moves in a clockwise direction, for example, you hold the helix or spiral coil or a spring in your hand, and when it moves, the coil moves away from the observer. If it moves in the clockwise direction, then this is called right-handed helix. On the other hand, when this coil or helix or spring moves in the anti-clockwise direction, then this is called left-handed helix. Right-handed helices are present in proteins and left-handed helix or helices are present in DNA only. So, we have seen the skew sense in the 3 dimensions, whether right-handed or left-handed. But when we write on a paper, we do not have 3 dimensions, we have only 2 dimensions. How to represent the skew sense, whether right-handed or left-handed on a paper, is simply that a helix is having 2 sides. One faces the observer and other away from the observer. So, when the side facing the observer is drawn in this way, that it rises from the left to the right side, rising from left to right, this is drawn as right-handed helix. Whereas, in the opposite sense, when it is drawn on the paper, rising from right to left, then this is called left-handed helix. Theoretically, left-handed helices are possible, but in natural proteins with L amino acids, these are not observed. Therefore, in the natural proteins, only right-handed helices are observed. The other set of discovered phi and psi angles has fixed minus 120 for phi angles and fixed plus 120 degrees for the psi angles. The polypeptide with angles in this range acquires a regularly repeating structure of an extended strand. This is beta strand, as it was the second regularly repeating structure discovered. The beta strands hydrogen bond with other strands to form planar sheets. The shown beta sheet is seen from the top of the sheet. This is the same beta sheet with 5 strands as seen from the side. This is the same beta sheet again with 5 strands as seen from the side, but side chains displayed. The side chains in a beta sheet always face above and below the plane of the beta sheets. The side chains are never in the plane of the sheet. Many a times there may be a turn present to connect 2 beta strands. Therefore, 2 beta strands will then run in anti-parallel direction. The regularly repeating structures, that is alpha helices and beta strands, they are joined together through what we call loops. Loops are not having regularity, that is they are not having the repeating same mean phi and psi angles. For example, in alpha helices, phi and psi are same throughout the helix or the beta strand, mean alpha helix having minus 60, minus 45, beta strand having minus 120, plus 120. These two structure mean one helix and other strand is joined through non-repeating phi and psi angles amino acids. These are called loops, and these loops are not regular, mean they are not repeating, but they are ordered. Ordered mean that the phi angle for one amino acid present in one molecule will be the same in other molecule for the same amino acids. So they are ordered but not regular, mean amino acid first in the loop may have value minus 60 for the phi and minus 150 for the psi, and for the next it may be minus 120 for the phi and minus 120 for the psi. So these are not repeating, they are not same for both the amino acids. So this is what we call not repeating or irregular structures, as opposed to the regularly repeating phi and psi in alpha helices and beta strands. And secondly, these loops are present on the surface. They are labile amino acids, they are not static as the amino acids present in alpha helices and beta strands, which are buried inside the core. The amino acid side chains which are buried inside the core are static, they are rigid, they are having a fixed structure, they don't move during their lifetime. Whereas the amino acids present in the loop, they are labile, they can move for performing a structure. For example, in the active sites and binding sites of the proteins are located in the clefts which are present on the surface, and these residues are hydrophilic and they are capable to move, they are labile. So they move for performing a function. For example, abstraction of a hydrogen from one group and donation to another. So they will abstract, donate, abstract, donate. So in this way they are labile and they are able to move, so that they are able to perform their function. However, the loops should not be confused with random coils. For example, an unfolded protein. Now in unfolded protein, phi and psi angles for the same amino acid in two proteins or two protein molecules are not same. So they shall not be confused with the loops shall not be confused with the random coils or unfolded polypeptide chains, because loops, although irregular, mean the phi and psi are not same for each of the amino acid present in the loop, but they are same for the same amino acid present in two protein molecules or all the protein molecules. We said there are 100 protein molecules and we are having glycine as amino acid number 1 in the loop, first amino acid in the loop. If this amino acid is having a value minus 60 and minus 60 for phi and psi, then the same value will be present in all the molecules. Whereas in random coils, unfolded proteins, this may have minus 120, minus 120 in one molecule and minus say 90 and minus 60 phi and psi in other molecules. So they are not same in two molecules. So this is what we call unordered random coil. So loops are not regular repeating structure, but they are ordered, mean having the same phi and psi values present in each of the molecule for the same amino acid. The second level structures, that is regular secondary structures and loops for the whole protein, may be displayed through phi and psi angles in a special plot known as Ramachamdan plot. There are three regions in this plot. First is the favored region, that is most favored region and is enclosed with a yellow line in this plot. The second is allowed region and is enclosed from yellow to blue line in this plot. The region outside the blue line are disallowed region. The phi and psi angle values in yellow regions in Ramachandran plot are favored for alpha helices and beta sheets. The regularly repeating structures including alpha helices, beta sheets and beta turn shows cooperative interactions to achieve these secondary structures. The phi and psi angles within yellow to blue region are allowed for amino acids present in loop regions. The regions outside blue are disallowed but allowed mostly for glycine residues represented with squares in the Ramachandran plot. Sometimes several secondary structures show orderly arrangement called supersecondary structure. A commonly occurring structural motif, helix turn helix, is found in many proteins such as lambda repressor of bacteriophage lambda, which regulates gene expression through binding to DNA. The supersecondary structures such as helix turn strand or helix turn helix should not be confused with the helix loop strand or helix loop helix, as the latter connects two regular secondary structures through an irregular loop. Dear students, we know the classical experiment conducted by Enfinson in the year 1973, where RNase, that is ribonuclease A, was unfolded by adding 8 molar urea and beta-mercaptoethanol. And when urea and beta-mercaptoethanol was removed from the enzyme was renatured. That is, upon addition of the urea and beta-mercaptoethanol, the enzyme was not functional. Therefore, we can say that it unfolded from its natural conformation, whereas after the removal of the urea and beta-mercaptoethanol, the enzyme folded back to its natural conformation and was able to perform its function. This shows that the information required for folding the sequence, that is the primary structure, into the three-dimensional tertiary structure is present within the sequence itself. That is the amino acid sequences. When they are polymerized on the ribosomes to synthesize proteins, they know how to fold themselves into the functional structures. And this folding is mainly through the hydrophobic interactions to pack the side chains and the secondary structures within the core. The secondary structures are having electronegative oxygens and nitrogen, but they are neutralized through hydrogen bonding of the main chain. And the side chains which are hydrophobic, they pack together, so that the packing force is mainly hydrophobic and the water is excluded from inside of the core. And secondly, in case a hydrophilic residue is to be buried within the core, then that has to be neutralized. For example, let us say that there is an aspartate which is having negative charge. If this aspartate is to be buried within the core, then it is to be neutralized first, and this can be neutralized with a positively charged residue such as lysine. Mean a negatively charged residue, aspartate, interacts electrostatically with positively charged lysine. The charges are neutralized and therefore they go inside the core of the structure to be buried in the hydrophobic core, because the core is hydrophobic, where the surface is hydrophilic. So the residues which are present on the surface are hydrophilic, and the residues which are buried inside the core, they are hydrophobic. Let us say that there is an alpha helix which is present on the surface. One side will be facing the surface, other side will be facing the hydrophobic core. Therefore, the side chains which are facing the hydrophobic core, that is buried inside the core, will have hydrophobic residues, whereas the side which is facing the surface will have hydrophilic residues, so that they can interact with the solvent, that is the water. The water is excluded from inside the core, because if water is allowed inside the core, then water molecules are labile molecules. They will be interacting with one side chain at one time, and at the next moment they will be interacting with some other chain. This will give the lability to the side chains which are present within the core, whereas water is excluded, so that the side chains which are present within the core, they are static, they are rigid, they provide a rigid scaffold for placing the hydrophilic residues which are labile present on the surface. So this provides the functionality, mean the core provides the scaffold, and this scaffold is rigid. This scaffold may be repeated in many proteins, for example in folds, what we call folds, beta barrel fold or Tim barrel fold. So that fold provides the static structure, whereas the moving loops or labile loops present on the surface or the hydrophilic residues present on the surface, they move with interactions with solvent molecule, that is water molecule, and therefore they are able to perform their function by relocating themselves for performing specific functions. The extent of hydrophilic surface can be compared with the hydrophobic patches on the globular protein using protein structure analysis software. Open a globular protein, display it in three-dimensional solid spheres, and color residues according to their type color, that is acidic as red, basic as blue, polar as yellow, and hydrophobic as gray. This shows that the majority of the residues exposed on the surface are residues which are either acidic, basic, or polar, with minimal hydrophobic residues on the surface. Dear students, we have seen two regions in the protein structure. The first is the core, the tightly packed static core, and the second is the surface which is labile. The tight packing is because of the exclusion of water, and in addition this tightness is provided by the formation of the disulfide bonds between the cysteine residues. These cysteine residues, they further make the scaffold static, so that the core is a rigid structure, and labile surface is present, but this surface is not random. So we have seen that the surface is ordered, mean that the amino acid having phi and psi in one molecule will have the same phi and psi in another molecules. So therefore in this way, although the structure is mobile, but it is not random. So this provides us the clefts on the surface which are present having active sites of the enzymes or the binding sites of the receptors. For folding, a contiguous single polypeptide chain is required. This may be full length primary sequence or a contiguous part sequence, that is subsequence of the full length sequence. The contiguous sequence, subsequence must be able to fold independently of the rest of the chain. This independently folding contiguous sequence is known as a domain. The protein shown in the slide has two domains in the tertiary structure. The domains have functional significance and are conserved during evolution. Different domains may be combined to create unique functionalities for these proteins. Each domain evolves for a specific function. Frequently, different secondary structures or different structures present in all proteins can be arranged in a limited number of combinations, each giving rise to what we call a specific fold, such as beta barrel fold, beta prism fold, Tim barrel fold, beta sandwich fold, beta propeller fold, and many more. A fold may be shared by different classes of enzymes, such as Tim barrel fold shared by an isomerase and a lyase, as in triose phosphate isomerase and enolase. Proteins having more than one subunit are said to have fourth level of structure, that is the quaternary level protein structure. This is achieved through non-covalent interactions between the subunits. This is significant to achieve regulation of the functions such as allosteric regulation, and hemoglobin is the representative example, which shows sigmoidal binding of the ligand oxygen. The protein shown here is a bifunctional enzyme having kinase and phosphorylase activities, with 3 subunits shown as ribbons interacting through non-covalent forces for allosteric regulation to modify the bound substrate protein shown in the slide in cyan-colored wire. Dear students, in this module, we have seen the hierarchical folding of the proteins having 4 levels. At the first level, we have seen the formation of the peptide bond through covalent amide bonds. At the second, that is secondary level, we have seen the folding of the main chain into alpha helices and beta strands, which are extended strands. Then these 2 secondary structure levels, they are joined together through what we have named as loops. The secondary structures which are regular are alpha helices and beta sheets or beta strands, and which are not repeating phi and psi having values, they are loops. Then these secondary structures, they are tightly packed within a core into 3 dimensions so that it has a surface which is labile, and this is the surface which is having the functional sites like active site of an enzyme or binding site for a receptor. Sometimes you see the secondary structures may have defined pattern, for example a helix, then a beta term, helix. This is called super secondary structure. Then the tertiary structure subunits or the independently folding domains, they interact with each other through non-covalent interactions and form quaternary structures, and through this quaternary structure the main regulation is achieved, that is the allosteric regulation. So the enzymes which are having quaternary structure, they have evolved for the regulation of their function. For example, hemoglobin, which is having 4 subunits, is a allosteric protein. I thank you all for visiting EPG Pathshala. Thank you.